Here we go. We can title these notes genetic mutations, or better yet, gene mutations. Types of gene mutations. The first type that we'll talk about is a type of gene mutation called crossing over. And crossovers are pretty common. It works like this. If you have two chromosomes, this is one chromosome pair, and this is another chromosome pair. And we can tell that they're paired because they're joined together at what's called the centromere here in the middle, that little dot. Crossing over happens when a chunk of one chromosome gets broken off and attached to another chromosome. So what we end up with looks something like this. So there we have one chunk of a chromosome that has now broken off and attached onto this chromosome here. Another type of chromosome mutation is called a deletion. Deletion. And deletions are lethal. Usually. Deletions work just like the name implies, where a chunk of chromosome is removed. So missing information is usually something that is deadly. Here we have this chromosome that is missing a chunk. That has broken off and is no longer attached to the chromosome. Another type of mutation, and we'll see this type of mutation in Chapter 6, is called polyploidy. tetraploidy. And the normal situation is where we have 2n. That for humans, that means we have 46 chromosomes. And those 46 chromosomes divide into two sets of 23. 46 chromosomes. Well, there's another situation that is called, let me get this into a situation where you can see it. Whoops. There's another situation where, is it okay to turn the page?
Yes, it is.
There's another situation where you have triploidy, and that's 3n, or 3 times the normal amount, and tetraploidy, which is 4 times the normal amount. It occurs when one egg is being fertilized by 2 different sperms, or one sperm fertilizing a diploid egg. There is a 4th type, and we'll see this in chapter 6 as well, called non-disjunction. Non-disjunction. And non-disjunction happens when pairs of chromosomes do not separate. For instance, IE means for instance. If you ever see IE, it means for instance. If you ever see EG, it means for example. So if you have 2 chromosome 21s, two sets of chromosome 21, that's called Down syndrome. Down's syndrome. And you all have heard of that before. You might even know somebody with Down syndrome. It's usually not a lethal situation, but they do have certain genetic characteristics. There's another one called Patau syndrome. And Patau syndrome is cleft palate. How many of you have heard of cleft palate before? A few of you, okay. Cleft palate is where there's a, where the dimple is. It's usually like a super dimple, and the babies have a really hard time feeding and eating, and there's some other things that go on with that as well. There's also something called Klinefelter syndrome. Klinefelter syndrome is one instead of an X chromosome and a Y chromosome, which is a male, or an X chromosome and an X chromosome, which is a female. And we'll get into this in chapter 6 in greater detail. Instead of this, so this is female, and this is a normal male. That's a normal male and a normal female. Klinefelter syndrome is one in which the person has two X's and a Y. And about 1 in 400 births is this kind of a genetic situation where the chromosomes actually don't separate. And there's other situations as well, like X, Y, Y, which is a male, a very tall male. So usually when these chromosomes, the gender-determining chromosomes, don't separate, it's usually not lethal. So when you have extra of the normal amount of information, it's usually okay. However, when you have missing information, like in deletions or crossing over, where it's out of place, usually that's more serious. Okay? I think I want to stop there because I want to get going on a protein synthesis lab that's going to be really fun and give you a better idea of how it works.